Right, good morning ladies and gentlemen. If I have your attention please, it's time to get going. Welcome to this year's AHDB Agronomist Conference. It's great to see so many of you here. It's one of our flagship events of the year, so it's fantastic to get such a good turnout today. For those of you who don't know me, I'm Tim Isaac, Head of Arable at AHDB, and I will be your chairman for the next 2 days. We've got a fantastic programme for you covering the whole rotation. Today we'll focus on potatoes, tomorrow we'll focus on cereals and oilseeds. In response to your feedback, you'll have plenty more opportunities for interaction and discussion compared to last year, and we've made sure that all of the speakers have got the message about turning research findings into practical take-home stuff for you to take away today. So hopefully you'll see those improvements based on your feedback from last year. In order to give you maximum time hearing from the experts, you'll be pleased to hear, keep my input to the barest minimum. So on that note, let's get on with the day, have a look at the programme. So after the opening address at 11:15, we'll start session 1, which is on crop protection in potatoes. Break for lunch at 1 o'clock. We've then got your workshop sessions, and you've all pre-selected your preferences for the workshop sessions, more on that later. Session 2 will be crop nutrition in potatoes starting at 3:30. And then we'll wrap up day 1 at about 5:15. So for those of you leaving us after today, you should be out of the building by about 5:30. For those of you staying, we intend to meet in the bar over at the hotel about 6:30. Dinner will be served at 7:30. And I will be reminding you throughout today, we do start at 9:10 tomorrow morning, so don't get too carried away tonight. Right, bit of housekeeping before we get going. Fire, there is no no drill planned for today. So if you do hear a fire alarm, please make your way out of the exits. They're all marked safely down into the car park and follow further instruction. Toilets, if you haven't found them already, they were just through the double doors at the back by the exhibition area. Please fill in your basis and NROSO forms. We've got plenty of points for both days of the conference, so take the opportunity to get your points. If you haven't done so yet, the forms will stay out, so try and do them during the break. There will be some filming and photography going on. Please ignore it. It's just so that we can capture and record everything from the day. If you do have any concerns about that, please chat to one of the events team and we'll try and help you with that. There is a link to all of the slides that you should be able to find if you haven't had it already. So don't worry about trying to capture all the slides. They're all on a link available to you. If you can put your phones on silent, that would be much appreciated so we don't disturb the speakers once they get going. But if you're keen on social media and Twitter in particular, please make sure you're using hashtag #AgConference18 and feel free to tweet throughout the day as long as it's nice and positive. In terms of questions, we're going to take them at the end of each panel session. So if while you're listening to the presentations you think of questions, just make a note and we're going to pick them all up at the end of that session. All of the speakers from each session will come back up here and you'll have an opportunity to question them at the end of the session. So just save those questions there. I'll be putting it out to the floor, so there'll be the usual thing with roving mics, but we'll also be using Slido. So for those of you not familiar with Slido, you can access it on your phone. So if you pick up the Wi-Fi for the event venue, which is in your packs, you go onto slido.com, you enter the event code, hashtag #AgConference18, so exactly the same as the Twitter handle, and that will then allow you to ask questions. So we'll remind you of that when we get to the first panel session, but just so you're aware, questions at the end of panel session, either from the floor or via Slido. Right, I think that's plenty from me already. I'd now like to invite Sophie Churchill onto the stage. Sophie is the AHDB Sector Chair for Potatoes, and Sophie will give us the opening address. Sophie.
Well, it's a great pleasure to echo Tim's welcome to you all and to wish us all a very good 2 days. I believe in the room we've got a very healthy mixture of potato specialists. It's specialists, not geeks, which is what I will say to our cereal friends. We are specialists, but we're in the room with our cereals and arable colleagues, and we have a mixed 2 days, which I think is where we want to get to. My role is to be the team leader of the sector board for potatoes, and that means we're the channel between the levy payers who represent the industry and that long-term body which is protecting us against the risks for British agriculture, which is the AHDB. Generally, board members of AHDB are recognisable by misshapen ears because our main job is to have our ears bent as we go out and about around the country. And I hope that you would recognise me by my ears. Much of my job is out and about with occasional visits to Stoneleigh. There are other people with misshapen ears on the potato board who are here today, and a big thank you for their membership of the board and everything they do. So our purpose today is to explore research becoming practical agronomy for the whole rotation. And there hasn't been a year when this has been more relevant to us, and I don't need to remind you of the variability of the results of the year. In the potato world, we're still waiting to see how potatoes behave in storage after not just the dryness of the season, but also the high temperatures. This year, AHDB has reissued its storage guide, for example, and our aim is always to give timely advice on the things which are exercising growers, agronomists, and everybody in the industry. So let's just celebrate the expertise that we have in this room and in the presentations and the displays outside. And as Tim has said, it is never the case, is it, that we learn by sitting and listening to the expert and then picking up something, taking it back to where we are and plopping it down where we came from when we left at 6 or 7 this morning. Change doesn't happen like that. Never does, actually, but particularly at the moment. What has impressed me so much about the agronomists whom I've met since I began at AHDB is the perpetual curiosity, the perpetual digging around to see what is different with this grower, with this field compared with the next field, with this variety compared with the next one, this March compared with last March. That perpetual excavating of what is going on now, and therefore what advice you might give to the person for whom you work, or for whom you're supporting at that time. And that is so impressive. And my hope for AHDB is that we reflect that kind of continual curiosity and responding to how things really are now. And I just want to make one point that may not get mentioned for the rest of the couple of days, but it is the context of the AHDB board and how we work. And that is, we are absolutely exercised on what happens ultimately in the kitchen and in the bar and in the restaurant, where the potato, one of my favourite foods, where the potato gets consumed. We all in this room want there to be this quality crop and cereals and other field crops for the next generation and the one after that. And the market, people's taste, the quality of those vegetables is at the heart of everything we do. And let nobody be in any doubt that maintaining consumption is absolutely the heart of what AHDB does. But that isn't just about marketing campaigns. That is the quality of the crop. That is the variety. That is the work that you do with growers and in the industry to make something that people are actually going to buy and eat. And I think it's maybe a good moment for me simply to thank you for the intrinsic and vital role that agronomy plays in the whole of the supply chain and the crop, which is a vital British food for us and for our children and for our grandchildren. And if we think that's difficult, just to say, I come from forestry in part. My background is partly urban issues, and then I did forestry. If you think it's difficult to work for the market in food and potatoes, imagine having to decide what kind of species of trees you might plant that's going to be harvested in 40, 50, 80 years' time. But in forestry, there's a lot of checks and balances, a lot of deciding, probably here, this might be the one to go. And there's a bit of playfulness as well. Let's have a go with this species, equivalent to a variety. And I see that same kind of experimentation in agriculture, and it is all to the good. Let me also say something which will be in your minds, but I think we should just say it at the beginning of our conference. The train has left the station in terms of the products, the chemicals, the inputs that we will be using in a generation or two's time. It may be too simple to say we're moving from chemical to biological. It may be too simple to say that integrated crop management is the is a catchphrase for all of us, and that somehow everything will fall into place. But that is the context in which we're working, and those of you who've got as many approaches and as holistic a view as you can of what it is to do productive, sustainable agriculture will be the most valuable members of our industry going forward. This all came to mind to me the other day. Many of you will have been in a situation. You're in your parents' house, but your parents are no longer there, yeah? The house is now empty, and inevitably what happens next is that the radiator on the top floor springs a leak in the empty house. That's generally what happens. And brown liquid dribbles and surges over a 10-day period down to the ground floor level. And you get the radiator mended, and then you have to reconstitute the room. And for me, this involved trying to find in this house a Phillips screwdriver so that I could put the carpet strip back down. And I remembered that my father had an impressive large blue metallic toolbox in the kitchen, bizarrely, because whenever you're looking for saucepans, you suddenly come across this great smart toolbox under the knives and forks. It's very large. It was a bit as if an aircraft carrier had docked in a small Cornish harbour. I sat there. So I remembered this, and I went to the cupboard and invoked my father. Phillips screwdriver, please, Pa. Opened the toolbox, entirely empty, except for 3 rusty drill bits at the bottom. Closed the toolbox, put it out to go to the tip, and then was on a journey through a much more chaotic, complicated house with lots of different categories of things, and found, roughly speaking, the right Phillips screwdriver in his aircraft modelling cupboard, and got the carpet stripped back. And I did all this while also thinking about agronomy and the potato industry, and the phrase that lots of people say, talking about, we've got less in our toolbox. So our challenge for the next 2 days is to compress the toolbox, to find all the other things around it, which may not look like Phillips screwdrivers, but which together are going to take us into the journey into the future. When we go on that journey, colleagues, I've got no doubt that our skills to meet the challenges of the next 10 years are with us now. The only thing I'd ask is that if you're a slightly younger person in this room, you never start a sentence with anybody here saying, well, I've only been in the industry 2 years, or I'm just a grower, or I'm just this. We are all here as one unit with our expertise to share, and that's what we'll do for the next 2 days. And I liken it a bit to what's happening in the livestock sector. I'm on the main board of AHDB. The movement to make sure that antibiotic resistance doesn't worsen, the movement towards vaccination, the movement towards husbandry at a higher and higher level, all that requires a different approach, a different skill set, possibly different people into the industry. But we can do it, and there's the most exciting transformation that is underway in the skills and in the types of people who are going to be working. And AHDB brings us all together to do that. And if we did nothing else but that and give chances for conversation, we'd be doing very well. But we do more than that, and I just want to close with just reminding us of some of the things that have been going on and which make our work all the more useful. Tim and I just had a conversation about whether cereals and potatoes are joined at the knee or the hip or somewhere else higher up in the anatomy. Somewhere perhaps between the knee and the hip. It's very important that we are, because we're in the rotation. But there's also something very distinctive about potatoes. They are fussy, high value, volatile, and need a lot of attention, high maintenance, and very exciting to grow. So in that context, AHDB in the last year has, as I said, published a new storage guide. It has continually refreshed its guidance on PCN and blight. We'll hear more about that, particularly on sensitivity, fungicide sensitivity. We're doing a lot on the Brexit analysis of the costs of pesticides, challenges of labour, machinery, imports, and much more. And AHDB, of course, works on the wider business skills and how we can make sure that our businesses are resilient into the future. And Farm Bench works across crops and across enterprises and is a very important tool for all of us. What AHDB, of course, doesn't do is control meteorology, markets, or Mrs. May. So quite a lot of the things that exercise us at the moment are not up for us to control. However, it is here with you to help us ride the risks and steer our way through the volatility. Myself, with the board and with our colleagues, will be drawing more and more on the expertise of agronomists to help us in that journey. And whether or not you would say you're independent or linked to a company or a process, it doesn't matter. It doesn't matter. The fact that you're involved in agronomy is absolutely central to what we're doing. So let me just close by reiterating it is a great privilege to chair the AHDB Potatoes Board and to witness your curiosity and your perpetual working out what might possibly work in this place, in this field, with this variety, on this spot farm or wherever else. I wish you a very good conference and a very good season to come. Thank you.
Right, thank you very much indeed, Sophie. Gets us off to a great start. Right, we're now going to kick off with the first session of the day, which is on crop protection in potatoes. We've got two presentations on blight, and I'm very pleased to introduce you to our first speaker, which is Geert Kessel from Wageningen University, who is going to give us the European perspective. Geert, if you'd like to join us up on stage. Yeah, just on the green clicker.
Yep, thank you. So good morning, ladies and gentlemen. Well, my name is Geert Kessel. I'm a plant pathologist working at Wageningen University, mostly on potato late blight. So although the title is on a European perspective, it's mostly a Dutch perspective and a Euro blight perspective. So that's what, this morning I'm going to have a co-presentation with David Cook. So somewhere midway the presentation I will just leave and David will come in my place. So don't be worried, it's all planned. I'm going to say some things about late blight in the Netherlands. The pathogen Phytophthora infestans is a highly dynamic, and that's where the source of all our problems is. Then I'm going to turn to the Euroblight perspective, pathogen population monitoring. I'm going to show you some of the work that has been going on future ways to control late blight in the Netherlands, host resistance, IPM on a new level, decision support, precision agriculture. We have a covenant in the organic potato sector at the moment, and I'm going to share a recently published vision by our Ministry of Agriculture on crop protection and food production in general. Then David is going to take over. So let's start off with potato late blight. Most of you will be familiar with these symptoms. It's destroying the whole plant, including the tuber, if you give it the chance. In the Netherlands, we on average spray 12 to 15 times per season, and that is from emergence to desiccation. So we do not wait until crops have closed in the row or between the rows. The first predicted infection event is the first spray if there is a crop above ground. The basis for that is or was a weekly spray schedule, more or less. If the weather is better, then it can be a little bit a longer interval. If the weather is wet, then the interval is usually a bit shorter. But we're also moving away from that because we are finding more and more problems, especially when the crop is growing fast or with tuber protection later in the season. So more and more farmers and especially advisors are using decision support systems to know what's going on exactly, to know whether they need to spray preventive, curative or an eradicant, whether they need to spray at all, what the problem in a specific crop exactly is. So spray programs are coordinated over the winter. So advisors go visit their farmers, talk to them about late blight control, and also of course they're also selling the fungicides. And if you know some of the names here, you can recognize some of the companies. Mostly farmers are using block schedules in the Netherlands. So they spray a certain fungicide 4, sometimes 5, sometimes 6 times in a row, and then change to another fungicide. And a very common schedule would be, first block would be Revus, Infinito, for instance, in the middle block, and then Ranman in the final block. Revis because it is very good at protecting new growth, and Ranman in the last block because it is very good at protecting tubers. Now, in the last year or so, more and more discussion also in the Netherlands about these block schedules, and I think it is likely that we will see some modification of these schedules as well, favoring a shorter alternation of different fungicides, of different active groups. As I said in the beginning, Phytophthora infestans is the pathogen. It's highly dynamic, and this is the root of our problem. It's changing all the time. Once we know how to control it, it's changing, and then it's out of control again. The pictures you see here are from a study from some time ago, but they are quite illustrative. This is a summary of 10 years of Phytophthora infestans in the Netherlands. They have been, let's say, genotyped or fingerprinted, and all these vertical little things, they are individual samples. And as long as they are nicely aligned like this here, they are genetically the same or almost the same, and we call them a clone. And here, this is the most famous clone of them all. This is Blue 13, which you will be very familiar with. And if you look at the whole tree here, you can see that we have a big clone, Blue 13, some smaller clones here, 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 and a lot of genetic variation. So individual samples that are different from everything else. So the Dutch population, similar to the UK population, is built of a number of clones, 3, 4, 5 clones, and a lot of genetically more or less unique Phytophthora infestances. Here, these red samples, is a nice example. They come from the northeast of the Netherlands, where we have our starch potato area and where we also have oospore problems. So there's a lot of genetic variation due to sexual reproduction in this area. You may also know that we sample these using FTA cards within Euroblight, but I will come back to that. This is from last year, this picture. Here on the right, you can see that these are the clones that we find in the Netherlands. EU1, the orange one, Blue 13, and here in 2013, for the first time, EU37. The gray bar, the gray part of the bar, are all these unique genotypes. So we had a mix of 3 clonal lines and a lot of unique genotypes. And then you see that in 2013 we have EU37 for the first time. 2014 we have EU36 for the first time. And they start, well, expanding their part, their share in the Phytophthora infestans population. You see that they are growing quite fast at the expense of, well, EU1, pink 6, and here Blue 13 and these unique genotypes. If we look back, 2013, this is where we first found EU37. 2014, EU37 was still in its, let's say, area where it was born, expanding a little bit, but we also find here, this is the Dutch-German starch potato area, EU36 for the first time. And in the years after, they start expanding. 2015, 16 was an average blight year, and 17, which was a good blight year. You can see that they also moved into the UK. If we move back from the Netherlands to, well, Europe, Euroblight in particular, within Euroblight, they did an analysis of all kinds of control measures, and they made a list of how effective they were. And they came up with 5 best practices that were the most effective control measures for late blight. So here you have this list. It starts with crop rotation. It's normally used against soil-borne pathogens, but in our case, late blight can also be soil-borne. Crop rotation, a removal of primary inoculum sources. So those are the dumps, the volunteer potatoes, Seed potato quality can be here. And we still have a very active program on that topic in the Netherlands. Resistant varieties is our most effective tool. Unfortunately, we are not using it, or hardly using it, or using it in the wrong way. And I will show you some more about that a bit later. And then we come to, let's say, daily practice during the growing season. Fungicides, spraying fungicides, controlled by decision support. There's two, let's say, key issues with effectively spraying fungicides, which is, first of all, the timing of the application. If you spray after an infection event, the effectiveness will be much, much less than when you spray just before an infection event. So we try to time our fungicide applications prior to or shortly before predicted infection events, taking into account fungicide degradation of the previous spray or sprays, but also crop growth. And I will show you how important that is. Then we have a long, long list of fungicides, and within Euroblight they are separated like preventive fungicides, which all of the late blight fungicides actually are. But some of the fungicides can do a bit more. So some of them have curative capability. Mostly fungicides containing cymoxanil or propamocarb. So that's only a few. And the last category is eradicant applications, so stop applications. And those are usually mixtures of two fungicides, at least two fungicides with a very strong curative and a very strong antisporulent component. Now, just to show you what decision support can do for you, this is an example from Indonesia, actually. But I could have picked any Dutch location or UK location. Here they are implementing a 5-day spray schedule. 5-day, every 5 days. So spray here, 1, 2, 3, 4, 5 days later here, and again they spray here. Now this is a screenshot from the decision support system that we have available in Wageningen. It shows the weather data, relative humidity, temperature. The blue bars are rain, but most importantly, the purple bars are predicted or calculated infection event. So you will always try to spray just before a calculated infection event like this. So the top bar, the top colors, is the overall advice. Green, it's fine, you don't have to do anything. Yellow, you need to spray a preventive fungicide. And here, orange, if you spray just after the infection event, Phytophthora is inside the plant, so the fungicide also has to go inside the plant. So you need a curative fungicide. And, well, if this spray would not have taken place, this would be red. This would be stop or eradicant applications. Now this is a 5-day spray schedule, here, here, and here. And you can still see that it's advising to spray 3 days after this spray application. And that is because here, this bar is showing the remaining fungicide protection of the previous spray on the sprayed part of the crop. So as long as there is anything, a little bit of green here, it's fine. So this is basically the sprayed part of the crop is well protected. That's what it says. But then the next bar, this one, is showing this red block here. This is let's say the protection status of the new growth. The new leaves that have emerged after the previous spray. So here, white basically means fine, 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 and then here, not so fine anymore. So this is the reason that this spray advice is taking place. So the new growth is triggering a very short spray interval here. Moving to pathogen population monitoring, this is, well, we don't have an official program in the Netherlands, so it's not supported by government or grower organizations. What is taking place is done from Euroblight. Euroblight is a combination of scientific partners and industry partners and is focusing on population changes. Euroblight also does fungicide efficacy trials and also the producers. They are monitoring within their product stewardship programs. So those are the 3 activities taking place in the Netherlands. And as an example, for instance, Green 33, well familiar to you by now, was first found in the Netherlands in 2009. And there was a lot of debate. Is there something going on? Yes, no, yes, no. And then in 2011, we did a trial like this. This is an efficacy trial with a lot of different treatments, but you can see the fluoxetine plots sticking out here. And this finally convinced the industry that something had to be done. So in 2012, the label of these products was modified to include a partner fungicide and spray it only in that way. So then in 2013, we first found EU37. The press release on that was on 2017, when it was clear that EU37 was less sensitive to fluoxetine. And results like this, they are usually presented during the winter meetings that we have with our farmers and our advisors. And just to show you how quickly these changes can go, this is the Netherlands in 2008 with respect to Green 33. We never heard of it. And then in 2009, we found Green 33 in a number of locations here in these central polder areas. 2010, it was slowly spreading. 2011, it was all over the place. In the winter 2011-2012 is when the labels changed, and also when all let's say the press releases came out, so the farmers were informed. The result was that fluoxetine use was reduced, well, a lot, but that also had a big effect on the population. As you can see here, in 2012, the Green 33 population in the Netherlands was decimated. And that was simply because we took away the competitive advantage for Green 33. So it was a little bit of a wimp compared to the other clonal lines that were around. With respect to the future, a lot of research was done over the past decade almost on how best to use host resistance. The risk with when you introduce host resistance is always that late blight adapts and breaks host resistance. So we were looking at ways to prevent that from happening. And this is a result here on the left from one of the trials we did in an EU project, where we had 3 potato varieties or genotypes, Desiree, Sarpomira, and this one is actually a Desiree with late blight resistance gene added. On top of that, we did 3 control strategies. Either we did nothing, we did not spray, or we sprayed every week, which is the common practice, or we sprayed according to what we called an IPM 2.0 schedule, which meant that we could delay sprays on resistant varieties, so start much, much later, or reduce the dose rate on resistant varieties. And a little bit we could also do this on a susceptible variety like Desiree. So Desiree susceptible, resistant, resistant. And then here you see the number of sprays, the sum of the dose rates over the whole season, and the final percentage of infection of the plots. So if you grow Desiree and you do not spray, you end up with a dead crop, which is not surprising. If you grow Desiree and you spray every week, under the circumstances of this trial, with a very high disease pressure, we ended up with a 5% infection level. And with IPM 2.0, we could not do a whole lot on a susceptible variety like this. We reduced the number of sprays by 1, and we reduced a little bit the dose rate, but we ended up with the same infection level as the, let's say, common practice. The advantage of IPM 2.0 comes with the resistant varieties, highly resistant varieties. As you can see here, we have Sarpomira, and you can look at this column at the same time, A15. If we do not spray, we end up with just a little bit of blight, and even less here. Now you might say that's not so bad, but this is the first indication of change within Phytophthora towards breaking this resistance gene. So this is what we really don't want to see. This is the first Phytophthora breaking this resistance. And that's what we wanted to prevent. Now, of course, you can do that with a weekly spray schedule. You spray 12 times and the crop is clean. Hurrah. The same here. Crop is also clean, but I think you already get a feeling that 12 sprays is a little bit too much on these resistant crops. We could do the same by just spraying the last 3 sprays, and even with a quarter dose rate. I do not recommend spraying low dose rates on susceptible potato varieties, but on a resistant potato varieties, there is some, there are a lot of things that we can do there. So 3 sprays, in total 3/4 dose rate sprayed over the whole season, and the crop is clean. Also here. If we look away from the individual experiments and we look at the total number of experiments, which was 5, 3 in Ireland, 2 in the Netherlands, and we look at the environmental impact, we get a graph like this. So here is the susceptible variety Desiree again, Sapomira resistant, and the cisgenic resistant Desiree. If we spray weekly, this is our environmental impact points. The 3 colors are the 3 environmental compartments. If we use IPM 2.0, we select different fungicides. We use a little bit lower dose rate, so it's reduced by almost half. But the big advantage comes with the resistant varieties. We hardly sprayed anything here and almost nothing there. And these crops were clean. And as long as they were clean and are clean, there is no risk of late blight changing. What's also happening at the moment is, well, decision support. I already introduced it here. But also there's a lot of precision agriculture going on. So with respect to spraying, we are going towards, we are spraying only when necessary. Only where necessary. So this can be, for instance, sensors detecting certain symptoms. Not for late blight, but for some insect problems you can do that. And spraying only this area. And also always use the minimum amount. So this is where we come up with variable rate applications, for instance, for worm killing, but we're also developing these for fungicides. And here at the bottom is an example for worm killing. So we have a satellite image of a crop. This is an NDVI image showing how much biomass, how much viable biomass there is. If the biomass is not very viable, you need very little Reglone in this case to desiccate it. If it's very viable, you need much more. And so here, in this map, is a translation of this map into a Reglone dose rate. And it is much lower over the whole field than the recommended dose rate. And if this is applied, then the crop is still dead. So this is the way we are moving with respect to precision agriculture. Organic potato production is very small in the Netherlands. And they had problems over the past years with late blight, of course. And some of them were spraying copper fertilizers against late blight. So copper fungicides, actually, fertilizers in a very high dose rate. And that was a big problem in the media. It's done in grape production very commonly, but in potato it is seen as a problem, and it doesn't fit with the organic thinking and the philosophy behind it. So they came up with a covenant early this year with farmers, breeders, trade, and also very importantly retail. And they more or less agreed that they only want to grow robust/resistant varieties by 2020. There's a number of late blight resistant varieties now on the Dutch market. Problem with them is that they are single resistance gene varieties, so they are kind of vulnerable. But this is where they are going in the near future. So there's a number of names here. And you can see some pictures from where the agreement was signed. There's a similar agreement now in Belgium, in Flanders, and in Germany they are also talking about going this way. This is my last slide. This is what our Ministry of Agriculture recently published. Their vision for 2030 on, let's say, Dutch agriculture. They basically concluded that current methods are unsustainable. They've been focusing on reducing costs, and they want us to move away from that and move towards reducing inputs and losses. So with respect to crop protection, they say that it has to be preventive, apply only when necessary, when really necessary, integrated pest management, of course, moving towards zero emissions, zero residues, including resistant varieties, so breeding, soil water management, biocontrol, low-risk compounds, etc., etc., etc. So this is quite something. It's going to be a big change. The reaction or the response in Wageningen was, the goal is to go to circular, climate neutral and nature inclusive agriculture. And the response in Wageningen was, that's very nice, but how? We don't know. So this is our challenge for the future. And well, let's see where it gets us. These are some of my conclusions. Yeah, I think you can read them for yourself, and I can invite David to the stage to take over from me.
Yeah, thank you very much indeed.
That's perfect.
Right, thank you very much, Gert. Great start for us. Just a quick reminder to keep your questions back for the panel session at the end, okay? So don't lose them when you're thinking of them as the presenters are talking. Our second speaker on blight is David Cook from JHI. We work very closely with David, so I'm pleased to hand over to him to give us the UK perspective. David.
Okay, thank you. Okay, thank you very much, Geert, for the excellent introduction. It's very nice to be able to follow on and from someone else talking about the Dutch population, and rather than me doing it for a change. Yeah, so I want to run you through a description of the population change in 2018, talking about linking to Euroblight, some work on aggressiveness from a project that I'm involved with, IPM Blight 2, which is a little bit confusing. It's distinct from the IPM Blight 2 project that Geert was talking about there. He's been leading the way, but the Dutch, unfortunately, were not part of that funded project. And then obviously, lastly, to talk about the fungicide sensitivity testing. So, yeah, I don't need to tell you that 2018 was a very unusual season. Clearly, in terms of the blight monitoring, we thank all those scouts who were engaged and were looking very hard for blight, but clearly the blight pressure was very low. So as you can see from the statistics on here, we only had 33 blight scouts were engaged, which is down from previous years. 40 outbreaks reported. I mean, the numbers in brackets are the previous season. And of those, we had 262 samples which came to the James Hutton Institute most of them via Ferra, from Murray Taylor in Ferra, and we genotyped 185 of those. So, and mapped them on the Fight Against Blight website, and the map is shown there. So the distribution was unusual, and I'll go into detail on that in subsequent slides. Just as a graphical reminder of, again, the unusual season, this is from Murray Taylor, who summarizes the data as it comes in from the blight scouts. A very low trickle of samples compared to the average over the many years that the Fight Against Blight campaign has been running. Why was that? Yeah, again, I mean, it's we did start with obviously a very cold start to the year with a beast from the east and an extremely cold and a more than average rainfall in April. So we did begin with some blight outbreaks on coal piles in Kent in this case, but I'm sure blight was active in other places as the temperatures did finally start to rise in April. But clearly what happened next was going to suppress late blight in almost any circumstances, as the rainfall averages for June and July, really just meant that at least you had to worry less about late blight, which I'm sure was very welcome. But you had plenty of other worries in terms of irrigation, I'm sure. However, there is some localized rainfall, and there were issues, as we went through August and September. There were circumstances in which blight did rear its ugly head, and from just anecdotal evidence from talking to scouts, there were occasions where blight, came along, and despite the lowest blight pressure year out, blight could actually be a problem in some circumstances. And I think that does partly reflect on new aggressive lineages, which were just able to sort of catch us out under those circumstances. because it was more active than expected. So we'll come on to some detail of that. So this is the chart showing the change of population, which you've all now seen for many years, but I really feel I still ought to just describe what we're seeing here. Is the coloured blocks are the clonal lineages of Phytophthora infestans, representing the ebb and flow of clones. So clearly the genotype 13 has a very rapid change. Genotype 13 itself has been declining, and this year is now down to 6%, which is the lowest it's been for many, many years. The grey block at the top is the other recombinant forms of blight. So those are blight types which are found rarely and usually only in a single crop in a single season. The clones we're interested in, I suppose, at the moment, 6A1 has been dominating, and indeed in 2018 still made up 46% of the population. So it's still there, it's still aggressive, it's not difficult to, it's still difficult to cope with. However, I suppose the headlines do tend to be taken by the new lineages. So we've got here the 37A2, which was also prevalent in 2018. 36, I would caution against, it did go up from 2 to 18% of the population, but bear in mind that the population sample was considerably smaller than in previous years, so you are always going to get sampling effects. We had a lot of samples from some areas, and I'll show you where those areas are, where we had late blight and a lot of 36A2. But the key point, I suppose, is that 36A2 did comprise a lot of, was the genotype found in a lot of outbreaks in your crops in this year. Okay, so adding some detail to that in terms of spatial work. I would stress this is actually a chart where I'm excluding 36 and 37A2, so I'm just talking about the other clones. And this is a reminder of the 2017 population, so the coloured dots obviously relating to the clones in previous years. So 2017, if we flick to 2018, yeah, clearly, a very restricted number of outbreaks reported. And the chart here shows also the number of outbreaks compared to previous years. Again, just bear that in mind in terms of sampling detail. So we did, the take-home message from this is really that 6A1 was still prevalent. Everything down in the southwest here, everything in Wales, and everything up in Scotland was 6A1, apart from the occasional grey ones, which are these other types that I've talked about in previous years. So, and there were small amounts of 6A1 and 1A1 over there in Kent. Now, if we compare that to the 36 and 37 on the next slide, again, this is showing a sequence of three years, but focusing straight away on the 2018 data. The 37A2 was present in pretty much the same areas as it was in the previous season, so Shropshire up into Lancashire. I think those were new northerly findings on the west, but nothing occurs really across on the east, this area. This is primarily because there weren't many blight outbreaks, full stop. So thinking about 36, a cluster here in Kent, and further up on the east coast as well. So we are seeing 36 kind of establishing itself. It's slow, but we did see it in the same sort of areas last year. So I suppose 36A2 we're not finding it in the west, and the other main take-home is that 36A2 has also not been found in the north or the west yet, which does have implications in terms of management practices for next year. Okay, so moving on to just think about aggressiveness work. I mentioned this project that the James Hutton are involved with, along with many other partner countries. So we all contribute isolates from our collections. So the Fight Against Blight, AHDB funded Fight Against Blight campaign contributed isolates to, in this case, the group in INRA, Rosalind Corbière and Didier Andrevon. So they've done some aggressiveness testing on these isolates. So this is some of their results now. And again, take-home message here, the lesion sizes from these clonal lineages, the number of isolates tested is shown below that. So 6 isolates of 36A2. The lesion sizes of 36A2 were amongst the largest, but next to 6A1, which makes sense, it's been an established clonal lineage. And secondly, they also looked at sporangial production, so the number of spores produced per lesion. And similarly there, 36A2 was up there in terms of producing large lesions with many spores. So that does fit with field observations of its spreading and being aggressive. Only another couple of things to mention there, I suppose. I mean, 13A2 is still there, perhaps not the highest. The differences aren't really too dramatic on these slides, but bear in mind this is a polycyclic disease, so even a small difference amplified over many cycles can add up to a big difference. The other thing I want to say, 39, this brown bar is a tomato-specific lineage, which did cause some issues for tomato growers in greenhouses last year. I won't dwell on that now. And yes, in general, this supports the evidence from outbreak sampling. Okay, moving on now to what's been waited for is there has been a lot of speculation, I suppose, and concern. We're seeing these lineages spreading dramatically on a European scale. What is driving that? We've just shown about the aggressiveness. Response to fungicides is an obvious question. So this is some work that AHDB commissioned in 2018. And we wanted to test a set of fungicides from a range of the Fungicide Resistance Action Committee groups for their ability to inhibit late blight. So it's a laboratory assay. Normal provisos there, it's giving us a guide only. It's not a measure of field performance. And we tested a limited number of isolates belonging to the established 13A2 and 6A1, that should read, lineages, and the 36 and the 37A2 lineages. It was conducted according to the FRAC protocols and concentrations. And it was a protectant test. So what we're doing here is treating the leaves, putting them in their trays, 24 hours later, inoculating them with these different lineages. And you can see here, for an example, just a tray of results showing some leaves without lesions and some with large spreading lesions of a range of doses of one product. So what did we test and why? As you can see, the list of products, list of active ingredients tested there is shown. They were selected from different FRAC groups. That seemed logical to test across the range. And this list was tested as a result of consultation with AHDB and the agronomists, senior agronomists in the country. We couldn't test everything. The very fact that we've tested these is not an indication of any issues. It's just that it seemed like a logical range to test. Okay, so moving on. I want to start with fluazinam. We know there's been an issue with 37A2. It's just mentioned it, the paper's published. I mean, it's documented now that the 37A2 is insensitive. And I want to introduce that and talk you through this chart, because the subsequent charts are in the same sort of format. So what we have here are a range of the results from different genotypes with the lesion size shown here on the y-axis, and 13A2 shows you the untreated controls, all in blue, and the effect of these increasing doses in terms of the lesions. So immediately you can see that 13A2 and 6A1, we've combined these together as representatives of the old population. They're all sensitive. The lesions we got from those when they're treated with that were 0 or less, or around 0. 36A2, no, let's start with 37A2, because this is where we know there's insensitivity. So the lesions were this sort of size, and yet even up to 1,000 ppm when we're using in these assays, we were still getting lesions. So here we've got an example where we really do have insensitivity. We're at field rate, and still lesions are forming. And that was elegantly shown by that field trial plot that Geert showed for green 33, but for 37A2 it's the same. So this is what resistance looks like. Now coming back to 36A2, there's perhaps a slight increase. The lesions under the lowest doses of fluazinam are more prominent than in 13 and 6A1. So something going on there, but only at low doses. Lastly, just before I move on from fluazinam, this also matches zoospore motility tests. This is not the official Fungicide Resistance Action Committee test for this fungicide. In fact, its zoospore motility assays are. And the results from the zoospore motility assays matched those published by the group in Wageningen, and it really supports that as being an issue there. Okay, so moving on to the other products now. Now I've talked you through the way the charts work. Cyazofamid. Note here the rates we're looking at are well below the maximum tank mix field rate, and that you do have to bear that in mind with all these slides. What we're looking at is the, again, untreated controls with lesions of such size. When we add doses, these very low doses, 13A2 is well controlled, 6A1 is well controlled, 37A2 also controlled. Perhaps a slight increase in lesion sizes here under the lowest dose in 36A2. These doses are intentionally made so low, and that is to get EC50 curves from these. So we have to look at an effective range of concentrations over which we can see a dose response curve to analyze the EC50 values. I'm not showing the EC50 values at this stage, but I'm showing just the lesion size because that's easier to get your head around in many ways. Okay, so the message here, I should also remember to say at this point, these results, Alison Lees, my colleague in James Hutton, has been the one who's been responsible for and conducting these experiments. I'm presenting the results for her. So, nothing much going on. There certainly isn't anything that we'd be calling remotely resistance in 36A2 in case of cyazofamid. Mandipropamid, similarly again, I'd draw your attention to the doses. We're, even at 10 ppm, which is well below field rate, we're getting complete control. There are no lesions here at 10 ppm. But one thing that does appear again from this is that the lower doses, the lesions formed at the lower doses of this product are larger for 36A2 than they are for the other lineages. Again, it's a minor effect, and I stress this is not resistance. Moving on to fluopicolide. You're getting the picture now. It's actually a very similar story for each of these products. Same sort of thing, 13A2, 6A1, strongly controlled, 37, perhaps the lesions are slightly larger at the lowest doses, and again, 36A2, just the lesions are just that little bit larger at very, very low doses. Again, 1 ppm compared to field rate of 500. When we look at the EC50 results that come through this, we do see statistically significant differences because of these differences in the way it affects the shapes of the curves. So although we see difference in EC50s, we're not talking about resistance. Lastly, propamacarb. Again, doses we're looking at here, I suppose in this case they're getting slightly closer to field rate, but the doses for this are very high, 5,000 ppm. And same sort of trend, some slightly increased lesion sizes at the very lowest, the lowest doses we're testing, so 100 and 250. You can see the lesions from 36A1 are slightly larger than those of other lineages. Had we not been able to discriminate the lineages, and we just put a group of isolates in here, 20 isolates, we would not probably have picked up on this. Because we can characterize these down to the individual clones, and we sort the data by that way, you can see these small differences. Okay, so some take-home messages from that. I want to move on. We certainly have supported the evidence for insensitivity to fluazanam. We knew that anyway. The good news is that this lineage has not moved north yet, so one is still able to use fluazanam in areas where we have not sampled it. We have no evidence of resistance to any active ingredient tested in genotype 36A2. We do, however, see these slightly larger lesions than other genotypes across low doses, and this is for all these active ingredients. So although we've just picked these products to test, we're seeing it across the range. So it does seem to be some sort of generic effect. We're not quite sure yet as to exactly what explains that. Presumably something related to aggressiveness of it. So we're not recommending any change to best practice. The follow the FRAC guidelines is the key message there. We do, however, need to keep considering this genotype 36A2 in particular. It is clearly more aggressive and can cause problems. So we're keeping an eye on this in future seasons. I'm sure we won't have such a low blood pressure year again, famous last words. So it would be interesting to see, how this develops in 2019. Lastly, I'd like to stress the fact that we're not the only ones who've been doing this fungicide testing work. The group in Wageningen have been intensively monitoring this. I know companies are doing their own monitoring, and in the IPM2 project, we also have been doing some testing. We are just, sort of first out of the blocks in releasing this data and discussing it. And what we plan to do is to sort of coordinate our data together with all the other research groups. And in early 2019, we'll come up with recommendations as a base based on a comparison of many other research groups. Okay, so yeah, we know it was an unusual season with low blight pressure, limited sampling. Clone 6 is still the most widespread. 37 remains aggressive, insensitive to fluazinam. Be aware of that in terms of your local area and whether it's been found yet in that across the whole country. Of course, absence is not a guarantee that it's not in your region, but we do seem to have some good indications there. 36, locally dominant and aggressive. No insensitivity detected. However, we do have that slight concern about the low doses. Remember again, your blight in your crops is coming from stuff which is kicking about quite often on your own farm or your neighbour's farm. So do keep an eye on those kill piles. If you control that primary inoculum, you're just making life so much easier in terms of managing the blight that comes into crops. So maintaining the focus on tuber blight management. I haven't mentioned tuber blight. We've had a few samples in, but not as many as previous years. And also, this is just one part of the results I'm presenting today. We have a lot of other IPM work that's going on at Hutton, which I haven't had time to go into. Okay, and lastly, my acknowledgements. My colleagues, Alison Lees, as I mentioned, was responsible for the fungicide testing work. Louise Sullivan and Debbie Powell have been isolating all the samples and helping doing the genotyping for many years. James Lynott is who's responsible for the technical work, doing all the fungicide testing. So he's been doing that work on the fungicides. And obviously my colleagues in Euroblight, Geert, who's here today, Moray, who provided us samples, Jens and Paul from University of Aarhus, who are responsible for the Euroblight database, which is necessary for showing all those pretty maps, and the Euroblight network, and funding from the Scottish Government, and of course from AHDB Potatoes. And thank you, I'll leave it at that.
Brilliant. So thank you very much to both Geert and David for a fantastic opening slot. Clearly blight is extremely important, and it's very important to us that we're doing what you want us to do. So in your packs you will find a survey about our current blight tools and investment. Can I please ask you that if you have any interest at all in what AHDB does for you in terms of blight, where we put our investment, resources, time, what sort of tools we're working on for you, please could you take the opportunity to fill in that survey and then either leave them on your tables or hand them in to us at the end. But it is very important for us to get this feedback so we make sure what we're doing is what you need in the field. Right, but for now we'll keep moving. Keen to keep to time and keep my teeth in. So our next speaker is going to be on PCN, another very important issue, and I'd like to introduce you to Peter Irwin from SARIC, and Peter's going to come up and join us on stage. Peter, over to you.
Hi, good morning. So I'm from the University of Leeds. We have a project to look at PCN, and in particular the tool that AHDB have that is used as an educational tool and an agronomist's tool to help calculate PCN populations and the potential damage that those populations would do. There's ourselves at the University of Leeds. See if that looks better. There's ourselves at the University of Leeds and colleagues at Harper Adams that are involved. We've got a lot of support from Andy Barker at Barworth Research, and the project is pretty much owned in the SARIC Club at BBSRC by AHDB Potatoes. So we're developing the PCN population tool. The calculator is a web-based educational tool. It's an advisory tool. It shows the implications of growers' actions on the level of PCN. However, it does require some improvement. And so that's what we've been charged with. Can we bring improvements to the current calculator? For those of you that aren't familiar with the AHDB tool, this is what it looks like. It's a web-based tool. There are various inputs. You can select your soil type, the population of PCN, if you know it. Before cropping, you can put in your cultivar. Do you know anything about your cultivar in terms of its tolerance, its resistance, or what treatment you're going to use? So there are a lot of variables that you can plug into the calculator to have an output on what the effects will be for the PCN population in your field. However, there are weaknesses with the model. The environmental parameters aren't adequate. The population estimates are uncertain around the economic threshold. There's an awful lot of question and variation and uncertainty around tolerance. There are a lot of resistant categories, and these are too numerous and not necessarily meaningful to the grower. There is a limited choice of nematicides and a limited amount of information on nematicides in the model. And decline rates, the rate at which the population persists in the soil, is not well defined for all populations. The consequence of these weaknesses are that it's not widely used by growers and managers. So our overall aim was to make improvements to this. Now we've got we are challenged with doing a lot of things, and I don't have time today to talk about all of them. What I would like to look at, what I would like to talk about is, I would like to talk about tolerance and resistance, but primarily about tolerance. And I would also like to talk about how these animals persist in the soil and what that means for farmers, because they're the two areas that our work has highlighted as being very important and very different from the current model. So comparison of field trial losses with those predicted by the current AHDB model and tolerance. And you can see that the darkly hatched bars here, the big bars, define field loss, but the smaller bars here are what the model is telling us. So there's a big disparity between the model and what the actual outcomes that growers are finding. And yeah, how useful is tolerance? With AHDB this year, we've done more trials. We've done more. The robustness of the tolerance trials that AHDB have done over the last 2 years have been incredible. But like any grower, kind of like they are subject to the environmental constraints that they're working in. And as we've already heard, doing anything in the last year, which is the driest years for 40 years, leads to some fairly peculiar results. We've looked at tolerance, and there are 9 different categories for tolerance, because in resistance to biotic pests, that's often measured on a scale of 0 to 9, and tolerance is measured on a scale of 0 to 9. And quite frankly, it's just far too many. It's just far too many. And when you go to the literature and you do a meta-analysis and you look at all the data in the literature and you say to yourself, are these categories all robust? It's quite clear that they're not. And so our thinking tried to simplify that, and we asked ourselves, well, could we get to a situation where we were able, we could have 4 categories. Could we have tolerant and not tolerant, and subdivide those categories into stably tolerant and not stably tolerant, and likewise for those cultivars that are not tolerant? And even when you try to simplify it down to that level, you go through the literature, you go through all the data and all the field trials that have been collected over decades, because trying to do it yourself in a couple of years, as we're finding, is just not possible. And you find that even then, tolerance is not sufficiently robust. So the question then is, what can we use instead of tolerance? Can we use determinacy? Oops, sorry. Can we use determinacy? Determinacy for potato growers is a very useful thing, because it's needed in terms of nitrogen application and other aspects of potato crop management. So can we use this determinacy instead of tolerance? And there's a lot of data already for determinacy. AHDB have ongoing projects to calculate what the best physical parameters are to measure determinancy and to measure the determinant values for new cultivars coming through. And this slide just shows, on a scale of 1 to 4, the different determinancy rates. If you compare the old tolerance rates to determinancy, they are indeed quite similar. There's two various outliers. You can see here for Saturna we have a tolerance of 4, determinancy of 2, and at the other end of the scale we have Vale Sovereign, which has tolerance of 2 and a determinancy of 4. But by and large, there's not a great deal of difference between determinancy and tolerance values. So the question is determinancy more robust than tolerance? Here we've plotted yield loss, and although they look like very small yield losses, they're yield loss per egg per gram of soil. So when you multiply it up by the number of eggs of PCN per gram of soil, these become quite substantial losses. And you can see that there's quite a good trend correlation between determinancy and tolerance. However, when you look more closely at the literature and you're doing your meta-analysis, you can see that those, the cultivars here that have a determinancy of 1, it's only based on 1 cultivar type. So these values are very ill-defined, and likewise, with determinancy values of 4, they're very ill-defined as well, only relying on 4 cultivars. However, in the middle for determinancy, values of 2 and 3, they're very well defined. You can do some very elegant academic mathematics, and you can plug in your determinancy values into the model, and you can find out whether your determinancy values are indeed worth looking at in a way that is greater than tolerance. And you can start adding extra things on top. You can look to see how much of the variation between what you actually observe and what you are forecasting in your model, how much of that variation can you soak up? And pretty much what you find out is the more you keep pouring stuff into your model, the more of the variation you can soak up. But from an academic mathematical modeling point of view, that's not very robust. And what you're looking for are the few things that have a big effect. So we've looked at a whole range of things to put in the model, and we can soak up all of the variation. But really, mathematically, we're not allowed to do that. We just have to look at the things that have a bigger effect. If we look at determinancy, we can find that we have an R-squared value, so the relationship between losses and what our prediction is, as it gets closer to 1, gets more and more robust. So looking at determinancy is very robust. And if we add other variables in, variables that we know have a big impact, we can look at rainfall. If we look at spring rain, we can soak up even more of that variation. And if we plot losses on one axis against determinancy in spring rainfall, we can see that crops that have a low determinancy value and where there is lower spring rain, there's an awful lot of loss to PCN. So at the moment, we've moved away from the old-fashioned thinking that tolerance is what you should be looking at for PCN, and we've looked instead at spring rainfall and determinancy, and we think this is an awful lot more solid foundation for predicting what that's going to do to the populations of PCN at your farm. So that was the first thing that I wanted to talk about. The next thing that I want to talk about, let's skip that. The next thing I want to talk about are decline rates. The animals persist in the soil, and this is just a nice fluorescent picture of a PCN. It's a female. She's gone through her life cycle on the plant. Her body wall's just fluorescing, just use some dyes here. The body wall is fluorescing green. The eggs inside her, she retains all her eggs inside her body cavity, and the eggs here have just been nicely stained red. And the longer this animal survives in the soil, the more these eggs will decline. They will become non-viable over time. And that decline rate is very important because it tells growers how often they can plant their potato crops. you can solely rely on decline rates, but most growers obviously rely on decline rates and the use of nematicides and resistant cultivars. But decline rates themselves are very important. The good news for us is that the individual animals are no bigger than they need to be. So a female will only grow to the size that she needs to be to contain all her eggs. There is no empty space inside the female. So if you measure the projected cross-sectional area of an individual female potato cyst nematode, you can make a beautiful correlation with the number of eggs that will be inside. And that means that in cross-sectional area of that animal is, you know when she was formed how many juvenile animals she contained. If you know how many years it is since you grew your potato crop, and then you extract the animals and you do break them open and do an egg count, what you have for your field is the decline rate for your field. So it's a simple way of working out decline rates at an individual field level. And I give lectures, and I commonly tell my students that of the two potato cyst nematode species, Globidira pallida and Globidira rostockiensis, that Globidira pallida, the decline rate is normally lower than that of Globidira rostockiensis. And so now I'm about to point out what I've learned is what I tell my undergraduates is complete nonsense. The influence, the duration of rotation, this influences the duration of a rotation course needed a very small difference in a decline rate. So a difference of 5% in a decline rate, 25% decline rate here and 20% here, can make a difference of 2 years in a farmer being able to put potato crops into the field. Persistent populations, those populations where the eggs don't decline and survive in the soil for maybe 2 decades, certainly need to be identified. And it would be really useful to be able to put individual grower decline rates into the model. So what decline rates have we found? Well, thanks to Matt Back at Harper and Andy Barker sending us samples, from a range of counties we have a mean decline rate of 9.4 with standard deviation of 5. But then from 2 different areas of Lincolnshire, we have decline rates of 20% and 25%. And remember, I've just said that having a decline rate of 5% can mean an extension in duration of potato cropping by 2 years. But the staggering thing that we found was that people that have put into their rotations a brassica are seeing decline rates of about 70%. And this was absolutely eye-watering to us because I'm involved in lots of biofumigation projects where the aim is to grow oilseed rape, to mulch it into the soil, and then the glucosinolates that come off that oilseed rape are having a nematicidal effect on the PCN. What this tells us is that those brassicaceous crops are having a detrimental effect on the PCN long before they're ever mulched into the crop. Now this figure here is a big figure, and that decline rate is a combined effect of the mortality induced by the biofumigation of the crop growing and the years since the crop, the last potato crop was planted. So it's decline plus the effect of the biofumigant crop. But 69% is an enormous decline rate. It's an enormous decline rate. And when we do some modelling to try and tease apart of that 70% decline rate, how much of it is due to the natural decline of not having a potato crop in the field and how much of it is due to a biofumigation crop being incorporated into the rotation, we estimate that having that biofumigation crop, could reduce rotational lengths by 4 years. So that's a massive effect. So the question then is, everybody collects their soil samples and they send them off to have their PCN measured. Is there a simple way that the decline rate for individual fields can be worked out, and so then when we go back to the model, we can put a decline rate in for your field. We're not guessing. Well, what we use in the lab is a beautiful Leica stereo binocular microscope, and there's just a picture of some cysts that was collected by that microscope that cost God knows how much money. You can simply draw a little box in one of these cysts and ask the computer just highlight everything that is that colour brown, and it'll highlight all the cysts in that picture. And you can measure their cross-sectional area. You can measure their cross-sectional area. You can buy calibration dots like this one here that show you how big something is. This one is from China. Those slides cost $12. Those slides cost $12. You can buy them in the UK for about £300 a slide. And so then we've compared the £300 calibration dots to the £12 dots from China, and there's absolutely no difference. We've also tried to take the image on an iPhone, and I'm sorry for iPhone users, but the image isn't quite good enough to be able to work out cross-sectional area. You can use a very simple microscope that costs around about £40 to take the image, and these work perfectly fine. So we think for the extension workers that are doing PCN counts, it's very cheap for them to be able to add on a measurement of decline rates for the grower. And they'll be able then to put your decline rate into the model to get a more accurate picture of what the PCN population is doing. And so that's just two of the aspects that we've been looking at. I'm sorry I don't have time to tell you about the others. If you catch me at lunch, I'll be happy to. Thank you very much.
Wonderful, thank you very much, Peter. Right, and our final speaker for this session is Simon Faulkner from SDF Agriculture. Simon's going to talk to us about developments in treating Rhizoctonia and black dot, and when Simon finishes, we'll move on to the panel session.
Good afternoon, ladies and gentlemen. So I'm going to talk about Rhizoctonia and black dot, and start initially with the black dot. Here you can see a typical photograph of the sclerotia resting on plant debris. So relevance, I should say, is more to the pre-pack industry. I suspect it's been a scourge of the industry for many years in the fact that in times of surplus, then you have quite a lot of downgrading because of the discoloration of tubers, and so much of the potatoes are bought on appearance. Strangely enough, complaint-wise, it rarely arises on the customer complaint radar, and it's not even in the top 5 complaints. It's commonly found everywhere where there's been a history of potatoes, and I suspect old fen soils, particularly Lincolnshire and Cambridgeshire, are some of the worst offenders. So black dot, here is the life cycle of it, and you can see down here the resting stage, if you like, during the winter on plant debris. And then as the temperatures come up in the spring, you get the spores being released and moving on to the growing plant. And finally, you get the plant dying and the black dot resting on that. The host for black dot, solanacea weeds, black nightshade, thorny apple, and also obviously volunteer potatoes. There is a school of thought there might be things like shepherd's purse and annual nettle, there might be a host, but I'm not convinced on that at the moment. So managing the rotation to keep your crops clean is important, especially through your whole rotation, not just your potato part of it. Identification, fairly typical picture of black dot. It's often confused with silver scurf. I think primarily because in the pre-pack world it seems to get lumped in together. They're both classified together on when they're looking at quality. But you can tell the difference, and a simple hand lens will tell the difference between black dot and silver scurf. You'll see under a small hand lens, you'll see these black lumps of coal that represent the sclerotia of black dot, and you'll also see the conidiophores like black threads for the silver scurf. And here's another typical picture of silver scurf. I suppose 20 years ago, silver scurf was a big problem, but I think with the advent of better cold storage and chemical treatment. We ended up getting that under control, but black dot came to the fore. So strategy-wise, understand the potential risk. Obviously, you've got to look at your land base, which is the most important criteria. Now you get a mixture of rented and owned land, and I expect a lot of potatoes are grown on rented land these days, trying to strive for cleaner land. Obviously, look at the history of potatoes on these soils. How many crops have they had over the years? What's the rotation length like on them? You can look at seed selection, but I do say that if you're striving for super clean seed, it's a bit needless if you're planting it into contaminated soils. Again, there is a DNA-based soil test that's available, and the AHDB had a project, 253, and they looked at this 2005 to 2007. And I think there's some Dutch work as well that was done 2007, 2008. And they found good correlation for the disease presence, but unfortunately I don't think the correlation was great for the infection. But I think that's more to do with the absence of adaphatic abiotic factors affecting the disease. Is there an opportunity to zone fields and test them when you're sampling for PCN? Considerations, again, variety choice. Customer is obviously key in most of this, but similarly, you don't want to grow a variety you can't grow well. Now, the AHDB NIO CUF work at Spot Farm East this year was looking at managing nitrogen, using a range of different nitrogen rates, and they're trying to see if there's any influence between that on black dot and also to bruising. Now, there is a school of thought out there that thinks that if you put a little bit more nitrogen on, you'll reduce your susceptibility to bruising. But I don't think the research is showing that at the moment. Obviously, delaying skin set, you run the issue of black dot forming on the skin. So avoid crops being left in the ground. Try and have a structured burn off where possible. Black dot on the infections on the skin can lead to sloughing of that skin at harvest time. And clearly, waiting for the skin set on that might never happen. So you have to take that risk and harvest it. But again, reducing your time for skin set is best. The hot summer of 2018 did lead, I think, to, in some sense, some premature senescence in individual plants. And again, it can come to the fore with these high temperatures. And this next slide just gives an indication of what the weather was doing here when you're seeing high 20 degrees and 30-degree weather, and rain down here, none within over a 48-day period, which is quite considerable. And this is probably a typical picture that a lot of people saw this year following this hot weather. So, summary. Good field selection, absolutely key, and I think that goes without saying. Try and get a long rotation between potato crops. Variety choice, look at varieties. Some of these new varieties have thin skins, and you wonder whether when they get infected with black dot, you get this sloughing much more. Seed selection, not such an important criteria on this, but it's still worth looking at and getting seeds sourced early. There are some soil treatments around. I mean, azoxystrobin has been around for a number of years now. It came out and it was going to revolutionize growing in soils that contain black dot, but in truth, it's not the panacea. It does help reduce the problem. Avoid crop growth stress, and again, manage nitrogen, as we alluded to earlier. If you're getting set skin in more than 3 weeks, you're obviously increasing your risk of black dot infection. Post-harvest, obviously, again, get rid of the moisture on those potatoes. Dry them as quickly as possible to pull that temperature down. Storing them below 4 degrees centigrade will help and reduce the development on it. And you tend to find that it doesn't develop further in store, it rather intensifies, and obviously detracts from the appearance of the tuber, which is what most people are buying them on for the pre-packed world. Biofumigation is another way to look at controlling the black dot. And the work has shown that there is some effect on black dot. But note a caution here, you need to have the right ITCs to be present. And you also need, in this particular disease, to have a high ITC concentration to make sure the pathogen is a negligible occurrence over a period of time. And work has shown that these to be up to 10,000 parts per million. This table here just gives you an idea of some of the ITCs that affect certain pathogens. And as you can see down here, this 2-phenylethyl ITC will affect black dot. I'd just like to acknowledge Fiona Taylor's work at SASA here that was funded by Barwell Agriculture for these next slides. So here you can see the growth line here, and then what the actual ITC has done. And up here, it's at 10,000 parts per million there. Chemical options. There are very few in terms of control here. You have one for the seed treatment, which gives you some reduction, and then you obviously have the soil treatment that we've alluded to already. But the chemical are limited on this aspect. So if we move on to Rhizoctonia now. Again, Rhizoctonia is found in most soils. And Rhizoctonia solani is ranged in a range of Astomostis groups, of which AG3 is the most important one for potatoes. I think AG2 is important, and that host is also sugar beet, so that's worth bearing in mind. It's a seed and soil-borne disease, and the problem is associated with both packing and processing crops, I think. The pathogen causes a range of problems. The usual things like stolon pruning and dwarfism. Aerial tubers can be formed in the leaf axils. You also get whitish mold found at the base of the stem just above the ground, and normally this is generally triggered by humid conditions. And finally you get the black sclerotia forming, which is the resting stage of the disease as the skins are setting. To the worst extent, you then can get misshapen tubers and growth cracking. And here's just one or two pictures just identifying that. Okay, factors for the disease. Obviously, black scurf on the seed is an important one, and it's an easy one to look for pre-delivery of your seed, ideally, and then post-delivery to see what levels are on it. Host weeds, interesting one is mare's tail. I'm actually seeing more mare's tail about these days, and I'm just wondering whether it's contributing to more incidence of Rhizoctonia and black scurf, and particularly on the silts. It's a very difficult weed to control. Stem canker favoured by cool, wet conditions following planting. Ironically, 2018 was a comparatively low-risk season. Any stress that slows emergence normally leads to Rhizoctonia. Deep planting with non-chitted seed. Again, it's a general debate that is had with growers. They feel that they should be planting deeper and deeper, but clearly it's not that. It wants to be planted the appropriate depth so we get emergence as quick as possible. Short rotations obviously lead to more Rhizoctonia. Intensive history of potatoes and delays to skin scare, all sort of things that add to problems with the disease. Treatment prevention, again very similar to black dot, field history. You need to understand what you're growing in, and how long have they, since they last had potatoes, and how many potato crops they've had. Planting conditions is obviously one to it. You want to wait for the best conditions, nice, warm, moist conditions. But I appreciate that in times where we've got growers growing big areas, you can't always wait for the ideal conditions, and you have to take a view on it. Seed quality, consider your seed options early. Make sure you go and select seed, because if you're not, you don't get the varieties you want, you don't get the quality you want. Variety selection, again, is important. There are susceptible varieties about, like Melody, Piper, Estima. Clearly, these are popular varieties anyway, and you get partial resistant varieties as well, the likes of Nectar, which is a very popular one now, panther and show palm, which is less so. Soil treatments, again, are limited. Seed treatments, you've got several options here. Again, you want rapid skin set so you can get harvesting early. And again, is there a problem looming with the loss of dicot? And finally, biofumigants is another option. So chemical options. Again, as you can see here, we have got several options, and I don't want to pick any in particular, apart from the fact they all have a place. But I would think the most important thing is to manage the timing and the application quality. Just a few observations on treatment, just chemical label changes. Could there be some ambiguities on timing around surrounding dormancy break? What are the best timings and sequence of applications? AI loading. The target dose from manufacturers is always higher than what we actually achieve in reality, which is something we need to look at. Liquid volumes, water, be careful we don't go too low. We need to have good coverage. Effectiveness of powders, an application on belt planters, how effective are those? Application equipment. I think there's improvements on this all the time. And then resistance and sensitivity, SDHI. Is that something we need to consider? Timing of application. There is some seed safety work being done independently where tubers have been dipped in the various seed treatments, and there's also been an application directly into the eye. This is just to see what the phytotoxicity is of it and what the emergence and dormancy break is like following this. And also be very careful pre-dormancy break and treating these tubers. Another slide on the same similar products. Just wanted to highlight this sort of there are one or two SDHIs there, and also with the new product, nematicide product coming on the market, which is also an SDHI, do we run the risk of any resistance? Just one for thought, really. A few slides of equipment that's currently out there. The one on the left is obviously for belt planters. The one on the right is direct injection system where you can put various chemicals on. So this appeals to customer choice, really. He can select whatever he wants, and it's as a good setup. And the last one is a sort of traditional roller table with the hood. So back to biofumigants again. Again, recent study shows that these ITCs have great potential to suppress soil-borne pathogens. The effect on Rhizoctonia, much more positive than black dot, but the ITC pathogen interaction is very specific, which is worth noting. And need to understand this to target the organism effectively. And the two specific ones for Rhizoctonia are methyl and benzyl ITCs have shown to have the greatest potential to suppress the potato fungal pathogens. Remember to screen the brassica to get the right ITCs from the right cultivar. This is back to that original slide I showed you, just picking up on the effects on Rhizoctonia with these two ITCs here. And finally, back to a similar grass I showed before, which was produced by Fiona, and it's showing here that at 125 parts per million, you get the thing to flatline, which is quite interesting. So in summary, there is continued work going on black dot Rhizoctonia. I think more work is required, and perhaps the review of application techniques of seed treatment. Clearly, I think the chemicals are good. I think the important thing is making sure we're getting the timings right, and obviously the application itself right. We're getting a continued loss of chemistry, so we need to be careful we preserve what we've got left. I think that biofumigants clearly have a great place, but certainly a lot more work needs to be done to encourage people to take them up. And obviously you need to make sure we use the appropriate brassicas to do this. And finally, back to basics, unfortunately, but good farming practices need tightening up to help with these diseases, because generally these are the problems that bring on the diseases. So land quality, availability, and I know it's a finite resource, so we are having got difficulty there. Seed selection important, and obviously husbandry methods as well. And on that note, I'll leave you with that slide. Thank you for listening.
Right, thank you very much indeed to Simon. That wraps up that session, so we'll now move on to the panel discussion. So if I could ask Geert, David and Peter to join us back up on the stage and take their seats with Simon, please. Now we have roving mics in the room, so if you want to go traditional and put your hand up, then the roving mic will come over and you can ask a question. Alternatively, use Slido, so get the Wi-Fi on your phone, go to slido.com, the event code is agconf18, enter those details and you can type the questions in and they will appear on the screen. So, gentlemen, are we all ready? Great. Right, okay, so we'll go to the floor first. Apologies, because I have a light shining straight at my head, so if you think I haven't seen you, please shout out. So we got any questions from the floor to start it with? Yeah, okay, so the one on the right first, please, Rob. Yeah, you, Rob.
Yeah, it's Rob Clayton from AHDB. It's a question for Geert. You made some optimistic comments about new varieties being available. I'm interested in the European Court of Justice this year made a ruling on cisgenics that wrapped them up with GMOs and made life much more difficult for licensing and for experimentation. I'm wondering where these new varieties are going to come from.
The new varieties are conventionally bred varieties, so there's no problem there. The problem is that they only contain a single resistance gene. So in the breeding programs now there is a lot of work going on in combining the different resistance genes, and that will allow us to have the, let's say, the full benefit of a strategy like IPM 2.0. So the GMO legislation is a little bit outside of this, although I did show you results on a cisgenic desiree. But it's just an example of a resistant potato. And so it's about the resistance, not the way we get them. But so far, it's only conventional breeding.
Okay, thank you very much, Geert. Okay, any more questions from the floor? Yes, so front middle down here, please.
Jamaican from Branson. A question for David, please. David, in your experience and with the situation with blight in Scotland in particular last year, do you feel it was wrong for agronomists to be advising growers not to use fluazinam at all in their sequences?
Yeah, that's a tricky one. I mean, the fact that we have not detected genotype 37A2 certainly suggests that that lineage has not yet moved north that far. The risks, though, of using fluazinam and the genotype being there but just below the detectable threshold, because no matter how excellent the scouts are, we do know we're not sampling every single field that has blight in it. That's a fact. I suppose that it was a risk-averse strategy, and I can understand why advisors are cautious. In hindsight, now we know the season was incredibly dry and spread of all blight was pretty limited. It probably would have been fine to use that product, but I can understand the risks. I would perhaps turn the question back and say, was there an issue with not using fluazinam that caused problems for growers? I suppose that's the question back.
Okay, Geert, would you like to add to that at all? Reflections from Netherlands?
No, don't think there's a need to add. Fluazinam is mostly used against sclerotinia at the moment, so it has its benefits there. But also it says on the label that you have to mix with something else if you want to use it in a late blight strategy. So stick to the label.
Yeah. Okay, great. Thank you very much. Yeah. Anything else on the floor? Yeah, so we've got two at the front here. So can we take Eric in the middle first and then over to, I think was it Mike? Was it you? Yeah, so start with Eric in the middle.
Eric Anderson, Scottish Army. A question for David, please. With respect to your data, David, or Alison's data, and 36A2, and given the relatively lower level of resistance shifts to both QIs and QIL fungicides, and also given the absence of any resistance shift to fluazinam, an inhibitor of ATP synthesis, does this indicate that we've got a potential efflux mechanism at work in 36A2?
Yeah, it's a good question. We've been pondering the issue there because I know I stressed the fact that at very low doses we're seeing lesions that are larger from one genotype compared to the others. And the fact it is across several products does suggest some sort of a generic feature of that genotype. And as you rightly say, there are processes within any living organism by which they can export any sort of toxin inhibitors, and that sort of efflux mechanism describes that. So it's just an ability to cope with any sort of toxin. And I suppose that is one possible mechanism that's going on there. It could also be, however, remember here we're treating the leaves, 24 hours later we're then inoculating. It could also be factors relating to the aggressiveness of 36A2 compared to other lineages. The infection efficiency, more spores are actually causing lesions. Those lesions perhaps are growing slightly larger. So they could also be related to the aggressiveness. Although the aggressiveness of 36A2, you will see from the untreated, wasn't always higher in those experiments than the other two lineages necessarily. So it's possible that's the mechanism. And I think one of the take-home messages that perhaps I didn't pull across there is that when you apply products in the field, obviously field rate is going on to the upper leaves. But as Geert nicely showed, there's two things happen, of course. There's new growth, there's areas where the product erodes, and there are areas that do not get the full dose. So over time, maybe those lower doses are present in the crop, and 36 could be active in those areas which are less protected. So I suppose the message is, if you're in any way concerned about application schedules being fully effective, it was a windy day, or the product just didn't get where you don't think the product got to where it should have done in the right dose, then bear that in mind in terms of scheduling for subsequent sprays. So.
The industry has used CAA products very commonly for rapid crop canopy development, protecting against blight at that stage. They work at that stage because they have historically been active at very low doses, because they are bound into leaf wax, and as leaf wax layer expands in the primordial tissue, they protect the leaf against blight for a few days. They need to continually be reapplied to be efficacious. So in that context, should we be worried about 36A2 for rapid crop canopy development and CAA products?
I certainly wouldn't recommend any concern over any particular product category compared to others. I mean, we've shown there isn't resistance. That is not to say that this isn't the beginning of a trend. We can't rule that out, but I'm certainly not advocating concern of any particular product group. I think just awareness of the fact this is an aggressive lineage, that even under circumstances that have previously controlled blight, the control failures could occur, but being aware of the fact that it's the aggressiveness and an erosion of product down to the very low doses that could be an issue. Just be aware of it in 2019. None of us can predict the future, but we'll, I think, keep aware of that and follow the FRAC guidelines. Yeah.
Okay, I'm keen to give everyone an opportunity and maybe give David a rest. Mike, have you got a question for another member of the panel?
Yeah, it's for Erwin. In terms of your tolerance assessment, you said you were looking at determinacy and spring rainfall. Those were the two factors that you correlated. What do you think is the mechanism through which those are affecting the response of the plant? So I'm interested in the mechanism, not the correlation.
Yeah, I don't know. I don't know how determinacy affects tolerance to PCN. I don't know what that mechanism is. How does spring rainfall affect PCN? I would just imagine that early spring rainfall gets the plants off to a fast start, and once you've got a vigorously growing plant, it's more tolerant of the infection than a plant that's got off to a slow start. Historically, there's a general conception that once the pathogen gets into the plant, and it's in the plant for about 6 to 8 weeks, that's when most of the damage occurs. But some really old historic data shows that, or suggested that, most of the damage actually occurs early in infection. So I think if the plants get enough rainfall early on and they get away to a good start, it offers then some tolerance against PCN.
Great, thank you, Peter. Right, shall we flip across to Slido, please, unless there's any other questions from the floor? No? Okay, let's see what we've got coming up. Right. Eric, I see you've been pretty busy with the questions. I'll take the top one because that's got quite a lot of likes, and it gives Geert an opportunity to speak as well. So does the Dutch strategy of blocking sprays actively place selection pressure on blight for resistance shifts? All blight in Great Britain was first found in the Netherlands. Don't take it personally, though.
Can I counteract that with Pink 6? I think that's a UK clone.
Sure.
Anyway, well, the first thing to say, of course, that the blocking sprays, the stopping sprays, they are not our first choice. It's an emergency measure, and yes, you are putting a lot of selection pressure on Phytophthora infestans, and that's exactly the reason why it is not our first choice. So if you want to destroy a product, use it this way. Okay. So that's why you should spray preventive all the time.
Okay, great, thank you.
Use the DSS.
I'm keen to, so we cover some of the other subjects as well. So biofumigants and PCN. Peter, are you happy to pick up that question?
Well, in a different project, which is the Horticulture and Potato Initiative that AHDB are involved in, BBSRC and AHDB had a project that ran alongside the Happy Project, and I was involved in the Happy Project. By far and away, the best biofumigate is issue 99. Different radishes work, but nothing as well as issue 99.
Thank you. Does everyone else know how to use Slido? Is it just Eric? Okay. We'll keep going then, but please feel free to put your questions in. Oh yeah, is there another one? Well, it's anonymous, I suppose. I'm just, Eric, I'm not sorry, I don't want to dismiss your questions, but I just want to give a few other people the opportunity as well. So let's pick up so will a reduction in use of fluazinim slow down the spread of EU37? Geert or David, do you want to pick that up?
Well, that's very difficult to answer because it's EU37 is less sensitive to fluazinim, but it's also highly aggressive. So if we compare to Blue 13, which is resistant to methoxyl, maybe that was the cause of Blue 13 emerging, but it certainly wasn't the cause of Blue 13 becoming so dominant. So there's more to EU37 than just fluazinim insensitivity.
Okay.
Yes, the comparison to the 33A2, which we could see certainly was not fit in the absence of fluazinim, and it declined quite rapidly. I imagine, I don't have evidence to this, but I imagine fluazinim use in Shropshire has changed dramatically, and yet it's still, 37A2 is still there, causing disease in subsequent years. If there are people still using fluazinim in Shropshire, then I would wonder why.
David, just quickly on that, have you picked up that bottom question relating back to that point?
Yeah, the appearance in West Midlands and what lessons can be learned. Ooh, it's a tricky one. I suspect it, given that there were no outbreaks in between, I mean, it's controversial, but it probably was imported seed. But I would not advocate stopping importing seed. The lessons, I suppose, we didn't know it was fluazinim resistant, so I suppose having that awareness of the traits associated with the lineage, I suppose to some extent we do rely on the industry doing their own testing in-house, and I know they do that. other groups do that sort of testing, but trying to get that information back to industry more rapidly would be one answer. And I know that's one of the questions on your form is to do with more monitoring and more rapid monitoring of progress throughout the season. That could be one answer, but it is quite challenging to respond within a season. I think is asking a lot from any research organisation. It takes a long time. So I'm not sure lessons to be learned. I suppose being responsive, getting that information as soon as we can is about all I can say.
Okay. Simon, you'll be pleased to hear we've got a Rhizoctonia question. So would shallower planting with later ridging up help with Rhizoctonia control?
Yes, I think it would. I think the important thing is to get the crop to emerge as quickly as possible. And once it gets away, then you've got every chance of avoiding these problems.
Peter, biofumigants, which ones are high in methyl and benzyl?
I've read that question, I can't remember. Somebody's going to have to email. If you email me, whoever put that in, you have to email me. I can't remember that off the top of my head.
All right, we'll pick that one up later. Michael, question at the bottom there. What are the institutes doing to address early blight, as in some areas now more prevalent than late blight, with very little tools for effective control? David, question around early blight. Do you want to pick that up at all?
Monitoring.
Yeah, I haven't worked on early blight myself, so I wouldn't like to build
there's a similar monitoring program going on in Europe on early blight as late blight.
Yeah.
Problems are different. We have two types going on, but there's also in early blight there's problems with reduced sensitivity to some of the fungicide groups.
So, and then we'll take the top question there. Is there potential for using bacteria products in furrow application to control soil-borne problems rather than relying on chemical? Peter or Simon want to pick that up?
Well, from my perspective, very little scientific information to suggest that that's possible.
Simon,
I think people are looking at it, but as I say, there's no data out there to support it.
Okay. Top question there is danger of overusing SDHI's various forms in potatoes, Anesto and All-Star and other crops. Comments on SDHI? Simon, do you want to pick that up?
I mean, I put that out. I don't know, but obviously on the list of chemicals, there's quite a few coming with as an SDHI, and obviously clearly we've got another one coming out as a nematicide. So just the question is, do you think there's a potential problem there? Does anybody think it's a potential problem?
Okay. Geert, what are the phenotyping results from Wageningen showing with respect to 36A2?
We've been running similar tests as David has. This was done in cooperation with a number of companies, but we are behind David, and so the results just come in. We still have to discuss them with the companies, so I cannot yet discuss them here.
Okay.
But we will come up with a joint advice over the whole range of experiments that have been going on in the Netherlands and UK and France.
Okay, another question there for you from Paul. Geert, is the low level of blight in Sarpomira not more likely to be as a result of the variety resistance breaking down with senescence?
Could very well be, yeah. But then still, blight that late in the season is a problem with regards to tube infection. So we still don't want it from whatever perspective.
Okay, Simon, the Rhizoctonia incidence question, FLN, role to play?
Sorry, which one? I think possibly they might have. I'm just not sure.
Okay.
If you know that, Peter.
No, you might get some damage caused by FLN that allows ingress of a secondary pathogen. That's a supposition.
Okay, and then I think we'll finish on the final question about EU37. Inevitably, the lunch question was going to come any second, so I preempted it. But why did EU37 suddenly appear in 2016 only in the West Midlands of the UK? Any reasoning behind that, David?
Speculation.
No particular reasons. I think I've already touched on that. I mean, I'm almost certain it was imported. Sorry, Geert, but there are other records of novel genotypes appearing, which the only explanation is either oospores or an imported seed issue. So I think there is an issue there. That's all I want to say, really. Yeah.
No, that's great. Okay, well, look, that's brilliant. Thank you very much. We got there with the Slido questions at the end. I think you got going a bit, and now we've got more than we need. So don't stop sending the questions for the rest of the day. Please send them in. If we don't get around to all of them in the panel session, we will go back over them. We will review them, and if we can get back to you on certain bits and pieces, we will. So don't just be put off because we haven't got around to doing it. So can we just thank the panel one more time? So yes, to answer the other question, we are now going to break for lunch, which will be downstairs in the lakeside room. After lunch, you'll be separated into your groups for the workshops. Just a quick note before we go on there, if any of you are wanting to taxi share to get home tonight or to get to a station or anything like that, we've put some little slips of paper in your delegate packs which allows you to book them, and we would strongly encourage you to group up with others. So the reason we put that up there is have a chat with others over lunch if you want to lift share home and hand those forms in. In terms of the workshops, you've pre-selected all of these. You should have, if you're just here today, you'll just have one colour on your badge, so that's easy for you. That tells you which workshop you're going to. If you've got two colours, because you're here for both days, it's the first colour that applies to today, and later on there'll be signage up and around the building, and there'll be plenty of staff on standby to help direct you to the right workshop. The workshops will start promptly at 2 o'clock. So please make allowance for that when you're having lunch. Go and look around the exhibitions, chat with others. That's what the idea there is for. But equally, please try and get to your workshops on time for a 2 o'clock start. They'll last about an hour. They'll be followed by a break, and then we'll be back in here at 3:30. So it's so lunch, then workshop, then break, and then we'll start session 3 at 3:30 back in here. Have a good lunch. Thank you very much.